Good afternoon, guys, and welcome to another revision tutorial for your GCSE revision. The topic that we are going to be covering today is the topic of organisation that you covered back in year 10. And as you'll see, we've got some questions here that we're going to have a look at together, go through, see if we can get some correct answers, deconstruct, and just generally go over things that examiners have said about these types of questions that might be helpful for you in your revision. So hopefully you've got a copy of this in front of you from the link below the video in the comments section. And as I've spoken to many of you about, make sure you work through it progressively. So maybe pause the video after this point, go back, find your revision guides, your work booklets, things like that. Have a go through the first couple of questions, come back to the video, play the video, see what the answers are, see what key things you've got or what things you might have forgotten, and then make any corrections you need, then repeat for another question, and work through it progressively like that. So let me just take a cup of coffee. So with that in mind, let's start with this first question. The one shows part of a deadly nightshade plant. Now, obviously, even in that first sentence, word deadly is something that we are going to be having to think about. Now, remember that rule that whenever there is a figure in a question, that figure there is to help you, it's to give you a clue, guidance to something that the question might be asking you for, and you try and find within that figure and interpret. So, got a leaf, and we've got some berries that are poisonous. So the question says, how the poisonous berries, so these are obviously important, help the deadly nightshade plant to survive? Now, this might be quite a common sense question in terms of just what you know outside of science as well as within science. But if berries are poisonous, obviously what they're going to do whenever an organism or an animal eats them is they're going to kill the animals. Now, obviously that might happen in one or two occurrences where an animal might be due to an environment and not know this plant. But if you've got a species that observe and learn over time that actually if they eat this plant with these berries, they die, then obviously that species is not going to eat it. So how's it going to help it survive? Well, it's going to stop animals. Now, you might want to be even more specific here and think about animals that only eat plants. Herbivores might be a word you might choose to use here. It's going to stop animals or herbivores eating it. Doesn't need to say any more than that. Yeah, but obviously we know that if they eat it, it'll kill them. Let's have a look at the next question then. So, which type of defense mechanisms are the berries? Mechanical, physical, or chemical? Now, poison, if you think about it, is something that is made of particular chemicals. So our defense mechanism is a type of chemical in the plant. Right, now, just turn second part of the plant, the second part of the plant, about the question. Figure 2 shows part of a gorse plant. Suggest how the gorse plant is adapted to defend itself. Now, same idea of the question, the previous one, figure 1. Figure 1 gives two clues. Figure 2 is giving us clues, but there's no labels. Now, we need to look and see what we can interpret here. Now, if you have a look at the question, what do you think those are on the stem of the gorse plant? They're spikes. Or thorns, another word we might use for it. So how do spikes or thorns or spines or prickles help a plant to defend itself? Well, same idea. The spikes are going to stop animals from eating it. Only one more question, so we don't need to say that it stops them from eating it because it hurts them when they eat the plant, and obviously would damage their esophagus and some of their digestive system. Those spikes might not be digestible. Don't need to go into that level of detail at all. It's simply just what are they going to do to defend it and stop plants from eating it because they're spiky. Good. Let's keep going. Green leaves of the gorse plant make glucose for the plant to use. I'm recording a video, kiddo, so I'll be out in a little bit, all right? Sorry for that interruption, folks. That was just my daughter asking me what I'm doing, because I'm doing this on a Sunday afternoon. So let's keep going. The green leaves of the gorse plant make glucose for the plant to use. What are two uses of glucose in the gorse plant? So it's that idea. We need to be thinking back to what do we know about glucose and what do we know about plants. Do we know glucose having anything to do with defense, protection of the plant? No. Do we know of glucose having anything to do with the absorbing of water? No. Releasing of minerals? No. Respiration. What's glucose used for in respiration? Energy, isn't it? Oxygen plus glucose gives us energy, and carbon dioxide plus water. So one of our options is for respiration. Now, I either want to start as starch. Starch, hopefully remember, is a polymer molecule made of lots of repeating subunits or lots of repeating monomers. And what is the monomer? Starch. What is the molecule that's joined together? Glucose. So we've got a second answer. Yeah. Brilliant. Right, let's keep going. So, a student wanted to show that the leaves of the gorse plant contain glucose. Well, the student crushed the leaves to extract the liquid from the cells and described the method the student could use to test the liquid from the cells for glucose. So what's the test for glucose? Now, this isn't related to plants, but we've learned about it in a different context. Not related to plants, but we're applying it to a new context. What chemical do we use to test for glucose, if you can remember? Now, if you've done a bit of revision, looked it up, you'll remind yourself that it's Benedict solution or Benedict's reagent. So describe the method the student could use to test the liquid from the cells. They've got this liquid, it's been extracted. What are we going to do with it? Well, 3-mark question. So 3 things we need to be looking for. Now, one of those things is the result. So this in itself is 1 mark. So our method only needs to be 2 marks, 2 points, 2 things that need to be done. So we don't really go into loads and loads of detail. Now we know we need to use different chemicals for test for nutrients like glucose. So our first mark is the fact that we add Benedict's to the liquid. Job done. Great. 1 mark. But what's different about glucose testing compared to things like Biuret reagent for proteins or iodine for starch or the emulsion ethanol test for lipids? Is one additional step. One additional step. Once it's been added, you need to heat the solution. Now, you might use something like a water bath. And that would get us our second mark, because what we're trying to get the solution to, a little bit of that wider science, is to above 65 degrees Celsius. Because then our reaction occurs and we end up with a colour change. Now, if glucose is present, what's the colour change? Firstly, what colour is Benedict's to begin with? Well, Benedict's to begin with is blue. Now, when it changes to that glucose is present, there's a couple of different colours we could possibly include here. The main ones to yellow, orange, or sometimes the phrasing you see used is a brick red. colour change in the solution. Those are 3 marks, and how you might go about just interpreting the question in terms of what you want to focus on. Right. Next question. The roots of gorse plant have bacteria that turn nitrogen gas into nitrate ions. Explain why nitrate ions are needed by the gorse plant. 2 marks. So remember that word explain is giving reasons. Why does the gorse plant take in nitrate ions? What are those nitrate ions needed for? When you think about the fact that nitrate ions are contain nitrogen, as you can see from the name, and when they contain nitrogen, nitrate ions give nitrogen to be used in nitrogenous chemical bases. What's the better word? Now, those nitrogenous chemical bases are what we find in protein, find in DNA, otherwise known as chemical bases. And that DNA, ultimately, as we've discussed before, is used to make proteins. So, nitrate ions are needed to make amino acids or proteins. So we've got one reason, but it's a 2-mark question. So what's the link between these two? What's the next step? What's the why? Why we need proteins to be made. So we need proteins to be made. Think about all the different things that they're used for. Which are needed for things like growth, new cells. Think about things like muscle tissue. They're also needed to make things like enzymes, hormones. Good. Next question. G. The roots of gorse plant can be infected by a honey fungus. And the honey fungus produces tiny spores on the ground. Suggest how the honey fungus spores travel from the roots of an infected gorse plant to the roots of a healthy gorse plant. So we're going from roots, and underground. What's one thing that's absorbed by the roots and travels underground? Why do plants have roots? Well, it's to absorb water. So these spores travel in water in the soil. Brilliant. Fantastic. So, finish that question. We'll go on to last couple. So that is apologies. Drug can be extracted from gorse seeds. So I've changed tact here slightly. So we're talking about drugs. We've got the word trial. We've got drug here. So we're starting to talk about drug trials. If you've looked at before in specification, linked into infection and response. So there's 2 things that doctors must test for when in a drug trial. What are they? Now, do doctors care about the appearance? No. Do doctors care about the taste of medicine? No, not number 1 priority. Do doctors care about whether the medicine can or can't dissolve? Might be taken in water or might be taken as pill? No, because you get medicines as both. So you've got these 2. Now, if you think about dosage, what do we mean by dosage? How much to take? Is that an important factor? Yes, it is. So they want to test what's the correct amount to take. Toxicity? Yes. because toxicity, what does that mean? Is it toxic? Is it dangerous? Is it poisonous? They want to know that before they give anyone the drug. So yes, absolutely. And the next question. In the trial, some patients would take tablets made from gorse seeds and some patients would take tablets made from sugar. We've talked about sugar pills before. Why do they take sugar pills and why do they take some of this medicine? So they can compare. They compare the results, what happens. Now these tablets have a specific name. Hopefully we should remember that it is a placebo. Because a placebo, no physiological effect. Why do they take these sugar pills? So they can compare and they can see any effects of mental factors of the mind. Good. Move on from that one. Now, question 2. Some data interpretation. Do enzymes and effective pH on activities of enzymes. So that's a really clear thing that we're looking at here. Effective pH on activities of enzymes. They help to digest food. So let's have a look at the question itself. Now, what's the optimum best pH for the action of enzyme Z? So we're looking at enzyme Z. There's a lot of information here. We've got 3 lines. This line, this peak refers to enzyme X. This line, this peak refers to enzyme Y. They're irrelevant. This, the only bit of data that's of any interest to us in this first part of the question. Enzyme Z. What's the optimum? What's the best? I mean best. It's the best relating to the activity of the enzyme. So best is when is it most active? So if we look at our shape, at what point is it most active? Is it when on our y-axis scale we're low down here, or when it's up high up here? So it's that idea that our activity is high. So it's this point here that's most active. We need a pH. So we have to find this point and then read again. Now if we look at our squares, we've got 8, 1, 2, 3, 4, 5. Between 8 and 9, there are 5 squares. So each of these squares represents 0.2. It hits the third one. So what we are looking for is 3 lots of 0.2, moving on from 8. So it becomes 8.6. Now, stomach makes a substance that gives the correct pH for the enzyme action in the human stomach. Name this substance. Repeat that question. Stomach makes a substance that gives the correct pH for enzyme action in the human stomach. What is the substance? Stomach. We know about a substance that's found in the stomach. We don't really know one substance. You've been taught it since year 7. Remember, we say that the stomach is acidic, don't we? To kill off bacteria, kill off chemicals as they move in. So, what's the name of that acidic substance? Clue for you. So, recall should be that it's hydrochloric acid. Now, which enzyme, X, Y, or Z, should work best in the human stomach? Now, we know from the previous question that the human stomach contains hydrochloric acid. So, if we're thinking about the human stomach containing hydrochloric acid, we need to think on here What's an acidic pH? Well, it's down here. It's a low pH, isn't it? So which enzyme works best in these kind of conditions are low pH? Well, it's only one of them. It's enzyme X, isn't it? All the way at the top. So in this question, just think about the fact that quite a few students, when they have done it, were able to read the enzyme Z's optimum pH for the graph of 8.6. However, quite a lot didn't know this, or couldn't apply the fact that we're talking about the stomach making a substance. The only substance we know that makes, we taught at GCSE, is hydrochloric acid. And that obviously, once you've got that, when it's hydrochloric acid, you need to be applying the pH idea to it, which is that it's a low pH, which leads us to enzyme X. So let's have a look at the next part of this question. Now, different parts of the human digestive system help to break down molecules of fat so that they can be absorbed into the body. Describe how, to gain 4 marks, you should refer to the enzyme, where the enzyme is produced, products of digestion, and any other chemicals involved. Remember that command word. Describe. What happens? Different parts of the human digestive system help to break down molecules of fat. So what are we describing? We're describing a process. Now, in this process, what do we need to include in terms of our description? We need to make sure whatever we describe in this process of breaking down fat molecules so that they can be absorbed into the body, we talk about these things. So it's a 6-mark question. Now, think about how we describe these things. Think about different areas which you know with regards to fat breakdown. And we've got quite a lot of possible marks that we can get. But remember, because it's a process, we want to include start. So what happens when it's first taken in, all the way to the end, when, as it says, it's absorbed by the body. So we need to get to that point. We need to include everything in between. So what else happens in between? So let's think about a few things and let's go through and bullet point what we know. Well, we eat fat. Whatever form it is, doesn't matter. Chocolate bar, we could just be licking butter out of a bowl, who knows? We're still taking in the same things. So what happens? Now, first thing, goes into our mouth. What type of digestion occurs in our mouth? Mechanical. We chew it. That's the first mark. Great. Where does it go after that? Well, it goes down the esophagus, goes to the stomach, and the stomach, we also know, is made of muscular walls. And it churns it about, breaking it down physically. It's got another thing that happens. Now, what else happens other than that mechanical digestion? We've got chemical digestion as well. So when we're trying to break down fat, we're trying to break down fats into 2 things. So they break down into fatty acids and glycerol. Now, if you think about it, we've just described what the products of this digestion are here. These 2 things. Great, fantastic. We've got that in. We've got a couple other things that happen. So what is it that breaks the fats into the fatty acids other than just mechanical digestion? Oh, it's enzymes, isn't it? Now, fats, if you remember, are more commonly known as lipids. What enzyme do we know that's related to fats and lipids that helps to break down fats into fatty acids and glycerol? It's lipase. So lipase is our enzyme. Great. Where's it made? Now we've got a couple of places it's made in the body that we looked at before. So lipase made in the pancreas and the small intestine. Help to break down fats. Great. But we haven't said anything about any other chemicals involved in the body. Now, we're not talking artificial chemicals, ones that are man-made and produced and put into the body. We're talking about some other chemicals produced biologically. What are the organs need fat digestion? And we have studied this. And if you look back in your guides, revision guides and booklets, you should hopefully remind yourselves that the other organ involved is the liver. So what's the liver got to do with it? Liver makes bile. Now, bile is two things. What it does is it neutralizes stomach acid, and as well as neutralizing stomach acid, it emulsifies fat, which makes it to smaller molecules. That increases the surface area. So now you've got lots of small molecules at the large surface area that are broken down by the lipase and the liver. Where are they absorbed into the body? One of the rules. Think about where you know about lots of different organs, what their functions are, and what they do. It's the small intestine. So the small intestine absorbs these small molecules produced by digestion into the body. Need a moment to have a look at that, and just while you're thinking about it, think about the fact that obviously the question requires you to describe how the human digestive system is able to break down molecules of fats they can absorb by the body. Now, it's a difficult question, and it's asking you to really display good knowledge of fat digestion. So thinking about those enzymes, making sure that you know lipase is associated with converting fat to fatty acids and glycerol. It's making sure you know that lipase is produced in the pancreas and all the small intestine. And additionally, bile is involved in the breakdown of fat. And as much as it's stored in the gallbladder, it's the fact that its actions is that it neutralizes stomach acid, and what you're trying to do is you're trying to make the molecules of that smaller before they come into the small intestine, and that's through the process of emulsification into tiny droplets of fat. We've obviously got the additional aspects of mechanical digestion by the teeth and the stomach, a bit like that. So don't forget those. But don't make the major mistake about misinterpreting the question and not limiting yourself to the remit of fat digestion. You don't want to waste time talking about carbohydrates, proteins, things like that. You don't get any marks for it because the question is very, very specific and just talks about that. So make sure you read the question to that level of detail that you can, and don't talk about things that it's not asking you for. Okay? Right. One more question. So, water and carbon dioxide are exchanged between the leaves and the atmosphere through pores called the stomata. Name the cells that control the opening and closing of the stomata. So if you remember about those diagrams of the stomata where you've got that gap, you've got that hole, but you've got cells around it. And these cells get larger and smaller, depending on the amount of water they've got in them. And we're just looking for the name. The name, if you remember, are guard cells. Okay. What's a useful plant in the transpiration stream? Describe two differences between transpiration stream and translocation. So QTs, what happens in transpiration and what happens in translocation? No similarities, just the differences. Now, the easy one to get marks for here is where they occur. So if you think about the fact that we know that transpiration occurs in one part of the plant and translocation occurs elsewhere, can you remember the name of the vessels that they're associated with? Part of the vascular bundle. One begins with an X and one begins with a P. Both occur in different vessels. So transpiration occurs in the xylem, whereas translocation occurs in the phloem. Now, another difference that's a really easy mark to get here is what is being carried in each of these. And apologies for the spelling. Transpiration, transport water, whereas in translocation, we transport sugars. Another difference you could get here is that in transpiration, substance only move upwards through the xylem, whereas in translocation, in the phloem, substances move up and down by osmosis and also using active transport as well. Okay, so a couple of useful differences there. Now, I've got a bit cold. Which environmental conditions would cause the rate of transpiration to be greatest in a plant? So remember, transpiration is due to the movement of water. What do we know about the movement of water? Water is moved by osmosis. What do we know about osmosis? We know that it's from high to low concentration to a partially permeable membrane. Now, if we think about our plant, we've got a high concentration of water. We know that rate refers to speed, how quickly it can. So we want it to occur as quickly as possible. That means it's to do with what's going on outside the plant that allows the plant to basically allow water to leave via the stomata. Now, in terms of our conditions, our answer is warm with low humidity. Firstly, why is it warm? If it's warm, what you do is you are increasing kinetic energy of the particles. They'll move faster. It's low humidity. What we mean by low humidity is the concentration of water in the air around the plant is low. So the fact is, we want to be moving from a high concentration to a low concentration of water. What we have here is that, which means because we've got a high to low concentration, we end up with a faster rate of transpiration because we've got a faster movement of water by osmosis. Good. Then that's part of our question. Figure below shows information about the mean width of stomata. dependent on the time of the day. Changes in the mean width of stomata in normal conditions are an advantage to the plant. Why? Now, if you think about what it's saying, is that it gets wider in the middle of the day and wider for a longer period of time. So gives us a wider opening and for more time. Why is that useful? Because the plant can take in more CO2. And that's for photosynthesis, isn't it? Why is that in itself useful? Oh, I'm doing the wrong question here, aren't I? Sorry, guys. What about? Really sorry. Thinking about the wrong question. Sorry. Right, let's come back to it. Change the mean width of the stomata in normal condition at advantage to the plant. Explain how. Now, how are these changes the fact that it closes, it opens in midday, and then it closes at advantage? Well, let's think about the fact when it closes. Why is that good? So they close at midnight. Why? Because it needs carbon dioxide that it's getting for photosynthesis, doesn't it? But there's no light during that time. We need light for photosynthesis, don't we? So what this also does is by closing it, means you've got reduced water loss from the plant. Whereas if it's widest at midday, why is it widest at midday? Because that's the point where there's most light photosynthesis. And if there's most light for photosynthesis at this point, that means because it's the widest, it means it can take in as much CO2 as possible for photosynthesis. Sorry about misreading that question there to begin with, because I was talking through the question below, whereby in the question below is read. Visualizing on time. Explain how the difference in the condition to help the plants survive in low atmospheric carbon dioxide. Let's come back to this point there, where we say that the graph tells us the stomata is wider for longer. If it's open wider for longer, get more CO2 going in photosynthesis. Take in more CO2 photosynthesis. If it does that, it takes in more CO2 for photosynthesis, well, it allows it to photosynthesize for longer and great detail as it possibly can, a great amount that it possibly can. You can see the two marks there, that if you've got an open wider, you can take in a lot more CO2 from that low carbon dioxide conditions, and it means actually you can do more photosynthesis. Good, apologies about that mix up on that one, but hopefully it gives you a little bit of an overview. Hopefully this tutorial useful. please feel free to come back to it as many times as you need. And obviously, all the resources are in the link below. And just to clarify, if anyone was looking at that first question, because I can't remember if I mentioned it, obviously plant defense mechanisms is part of triple science content. So if you're looking at it from a combined science point of view, don't worry too much about that. Okay, look forward to doing this again with you on the next one.